Hi everyone. This video introduces our MobiCom work, PDLens. Smartphone knows drug effectiveness among Parkinson's via daily life activity fusion. This work is done in collaboration with the University of Rochester Medical Center. Parkinson's disease is a brain disorder that will affect about 930,000 people in the United States by 2020. And is estimated to affect 1,238,000 people by 2030. According to the NIH, Parkinson's disease has four main symptoms, which are tremor in the hands, arms, legs, jaw, or head, stiffness of the limbs and trunk, slowness of movement, and impaired balance and coordination, which can lead to falls. Although there is no cure for Parkinson's disease, medicines and other therapies can often relieve some symptoms. The main medication for Parkinson's is levodopa, also called L-dopa. Nerve cells use levodopa to make dopamine to replenish the brain's dwindling supply. Usually, people take levodopa along with another medication called carbidopa, which prevents or reduces some of the side effects of levodopa therapy. In general, drugs prescribed for PD have three functions, which are 1. To increase the level of dopamine in the brain. 2. To affect other brain chemicals in the body. And 3. To help control non-motor symptoms. Here is an example to show how medication helps people with Parkinson's disease. In this video, the subject is asked to do the following activities: stand up from a chair, walk unassisted in a straight line, and come back. The video on the left shows the performance before the medication, and the video on the right shows the status after medication. We find that the subject can do much better after taking medication. However, there is a phenomenon called wearing off. Around 3 to 4 hours after a dose of levodopa, patients will typically start to experience changes in motor and non-motor symptoms, as the medication literally wears off and symptoms reemerge or worsen. Symptoms then typically improve 15 to 45 minutes after the next dose is taken. Wearing off tends to happen more frequently as Parkinson's disease progresses, and when it does, the control of both motor and non-motor symptoms fluctuates. As wearing off becomes more frequent, it may become increasingly difficult to control symptoms and predict when medications will be effective. This phenomenon is known as drug resistance. So our goal is to identify drug resistance in time, thereby adjusting drug treatment to improve the symptoms. An occasional clinical visit cannot let a physician fully understand the progress of Parkinson's disease. The in-home self-assessment sometimes does not work because the progress of drug resistance is so slow that people may not notice it in time. This is an effect called creeping normality. Thus, we are encouraged to propose a better solution to monitor drug effectiveness in daily life to assist the assessment in clinics. In this paper, we present PDLens, a smartphone-based end-to-end system to facilitate self-detection of Parkinson's disease drug effectiveness. It leverages the built-in smartphone sensors to collect digital Parkinson's disease biomarkers from users' daily life activities. These daily life activities are sensitive to dopamine levels in brains, and they are therefore responsive to drug intake. The proposed PDLens consists of a data collector, a data preprocessor, and a drug effectiveness detector. For the data collector, we leverage the smartphone's built-in sensors, which are the accelerometer, gyroscope, and microphone, to collect digital Parkinson's disease biomarkers, which are gait, balance, and voice, from users' daily life activities. In particular, gait refers to a person's walking patterns, and balance measures their falling risk when standing. For our drug effectiveness detector, we propose a ranking-based multi-view neural network. Convolutional layers and LSTM layers are adopted for feature extraction. Then, RankNet is adopted to compare the symptom severity at two different times, I and J. Weights in the neural network are not shared among each view, while weights are shared between the two branches of time I and time J. For the architecture of our convolutional neural network, we employ 4 residual blocks, each with 4 convolutional layers. The first residual block has 64 filters, and the number of filters doubles for the following blocks. We have a 7x7 kernel size for the first convolutional layer, and 3x3 filters for the remaining layers. For the architecture of our LSTM, the first LSTM layer has an input of 512x7 and an output of 64. Our second LSTM layer has an input of 64 and an output of 64. For the motor symptom score, we formulate the problem as follows. Given a pair of feature vectors with different timestamps, we adopt a function, called g, to map them to a numeric value, where xi stands for before drug intake and xi stands for after drug intake. These two outputs are then mapped to a learned probability via a sigmoid function. In terms of our loss function, with knowing the probability function, we adopt the cross entropy as a cost function to penalize the deviation of model output probabilities from desired probabilities. For a case study, we let S of ij equal 1 when xi is before drug intake and xj is after drug intake. We let Sij equal 0 when xi and xj are both from after drug intake or before drug intake. The equations can be seen on the screen. When the drug works well, the data collected from before and after drug intake will have different data representations. Min L1 and min L2 work well with this situation. However, min L1 and min L2 compete with each other if the medication loses its effectiveness. We collaborate with the University of Rochester Medical Center to enroll 81 people. Each of the participants in our study has been clinically diagnosed as having Parkinson's disease. This clinical trial lasted for 6 months. The participants were asked to perform 3 activities per day. The 3 activities were the voice activity, in which they would perform a 10-second long sustaining vowel. The walking activity, in which they would put the smartphone in their pocket and walk for 20 steps. And the standing activity, when they would put the smartphone in their pocket and stand for 30 seconds. Furthermore, we adopt the MDS-UPDRS, which is a standard clinometric assessment that collects wear-off information in daily life. We carry out 4 experiments to evaluate our system. The 4 experiments are 1. Evaluation of drug response detection. 2. Evaluation of drug effectiveness detection. 3. Understanding roles of drug effectiveness detector. And 4. system overhead analysis. For the evaluation of drug response detection, our goal is to measure the performance to differentiate between before and after drug intake. For each user, we adopt 80% of the data for training and 20% of the data for testing. This confusion matrix chart shows the results of drug response detection. Our system achieves 91% accuracy. Also, the recall is close to the precision, suggesting that we can differentiate between two stages without obvious bias. We also evaluate each single smartphone activity on drug response detection. We find that voice achieves a higher accuracy than gait and balance. The reason could be that vocal impairment is the most widely existing PD symptom in all stages, and drug intake can relieve this symptom. For the evaluation of drug effectiveness detection, as mentioned, we adopt the standard MDS UPDRS score as a ground truth. The figure on the right shows the CDF of scores. The higher the score, the worse the drug effectiveness. The figure on the right shows the correlation between the score in standard drug effectiveness test and that in PDLens. Although all the patients respond to drugs, the drug effectiveness varies from person to person, where the highest score is 0.72 and the lowest score is 0.2. Moreover, the score predicted by PDLens is correlated to that in the standard drug effectiveness test with a Pearson correlation coefficient of negative 0.36. This result meets our expectation that the participant with poorer drug effectiveness will correspondingly receive a lower score in PDLens. We further investigate that the Pearson correlation coefficient is negative 0.56 for participants who are medication onset for more than 2 years. Since drug resistance usually appears later after medication onset, e.g. after 2 years, PDLens can detect the occurrence of drug resistance. We then investigate if prior knowledge of medication onset time can assist PDLens to screen poor effectiveness participants. In particular, we define good drug effectiveness if a participant on average feels no or very slight uncomfortable, and define the remaining participants as poor drug effectiveness. The red dots mark the good drug effectiveness, and the green dots mark the poor drug effectiveness. This experiment is conducted with a linear support vector machine, SVM, classifier. We adopt a 10-fold holdout validation. In each fold, we randomly hold 15% of the data as validation. On average, we achieve 74.4% accuracy. For the system overhead analysis, we execute the model on smartphones and test the model footprint. The Google Pixel 2 achieves an average runtime of 1770 milliseconds, an average CPU percentage of 15%, and an average battery usage of 358 mAh. The Samsung Galaxy S9 achieves an average runtime of 1200 milliseconds, an average CPU percentage of 14, and an average battery usage of 488 mAh. The Pixel 2 is projected to use 1.06 mAh per day, which is less than 0.1% of its 2700 mAh battery. As for limitations and discussions, the calculated correlation R equals 0.36 between system output and score in drug effectiveness test is not very high. One reason for this could be that wearing off varies from individual to individual. Additionally, PDLens leverages a multi-model architecture for multiple activity fusion. However, it is difficult to quantify how much each activity contributes. Also, voice achieves higher accuracy than walking activity, and standing activity achieves a worse performance. One reason for this could be that vocal impairment is the most widely existing PD symptom in all stages, but the balance problems usually emerge in the late stages. In conclusion, allow me to summarize our work. We investigate that everyday activity fusion can augment drug effectiveness detection of PD. We design and implement PDLens, a smartphone-based system for detecting drug effectiveness in daily life. And we evaluate our proposed PDLens on a dataset collected from a daily life scenario. Thank you, and for more information, please read our paper in the conference proceedings.